Okay, well, here I
Okay.
am with uh, Stephen Gardner. Is that right, Stephen?
Steve.
Steve Gardner. He's the head of Precision Life. And boy, when I looked at what Precision Life was doing, my mouth just kind of gaped open. At least what they're trying to do. It's a very different approach. It potentially promises much quicker resolution of these diseases, or help for these diseases, um, and it's trying to do it all. It's not only is it trying to identify biologically based subsets, uh, but then it's trying to determine the mechanisms driving those subsets. Then it wants to find biomarkers for those mechanisms. And then it wants to find drugs that will move those biomarkers. And uh, that just puts treatment on a whole new level, on a much more specific level. Uh, and but I wanted to ask you. Um, so Precision Life is working with Metrodora and Decode ME on some large studies,
Yeah.
and we'll get to those later. But so what I noticed that really struck me is that you published two MECFS studies and you funded both of them. Um,
Yep.
so I'm really curious as to how you got interested in MECFS. And I have one hypothesis, and that's you chose the biggest, baddest, most heterogeneous, most difficult to understand, the yuppie disease, the disease of a thousand names, blah, 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 blah. And if precision life works in this thing, then it can work in anything. That was my kind. That's what I kind of like. I have no
Well, there's
idea how you came about
no,
this,
there's
but
there's a certain amount of truth in that. And I think you're absolutely right. Recognizing this was really heterogeneous and was going to be a big challenge. That's very much the problem that we as a company have set out to solve for complex chronic diseases. So, being a precision medicine business, usually they focus on oncology or rare genetic disorders, and the causes of those are relatively straightforward to pin down. It's usually um, mutations in single genes that cause those diseases. In things like ME, long COVID, and a variety of others, it's actually a variety of different impacts. So you have lots of different genes involved. You probably have multiple different mechanisms involved. There's an overlap with the immune system, there's an overlap with the environment. So all of these factors come together, and it just basically makes it a lot more complex to analyze. And that really is, I suppose, our secret sauce and differentiator is that we can deal with that complexity. Um, but the
so
other reason,
you're actually looking for complex diseases.
yeah, oh,
That's
absolutely.
that's your that's that's what you excel at.
Yes,
Yeah.
absolutely. And we're choosing
The other
those because
diseases don't necessarily need you.
there,
These
there are,
diseases really need you.
yeah, exactly. There are
Yeah.
plenty of other players in those other in
Yeah.
oncology and rare disease. They're very relatively well studied, and we can make much more of an impact in these other diseases. Um, the other reason was a slightly a slightly negative one, if you like, which is the recognition that these were not the same diseases as we were working with our biopharma partners. So, we can work on Alzheimer's, we can work on schizophrenia, we can work on ALS, and because we have commercial projects running there, we have to keep those results secret. We wanted some diseases where we could actually publish fully the results that we were generating, and
Oh, interesting.
this sort of came to a head in March and April 2020 when COVID-19 hit. So we sent everybody home like everyone did, and we're scratching around to see, well, everybody's sequencing the virus, and they're running these big studies. Nobody knows what's going on with this disease. is there something that we could be doing that would actually help this global program, this collaboration of the scientific community? And in May of 2020, UK Biobank put out the very first data set where we had the host genome, i.e. patients' genomes, and we could split them into those who only got very mild disease or asymptomatic, and those who had life-threatening forms of the disease. And that was really our first study. Back in June of 2020, we put up a paper on medRxiv. We found 68 genes, and we managed to show that we could correlate those to all of these weird and wonderful symptoms that patients were not really supposed to get from a respiratory disorder. If you
Mhm.
remember, way back then, we looked at obviously inflammation, viral replication, all of those things, but also lipid biology and the breakdown of plasma membranes, the senescence of cells, the neurodegenerative components. We saw blocks of genes for all of those. This was also, the period when everybody was looking at repurposing candidates. It was way ahead of the recovery study, the UK study where we found
Right.
dexamethasone. But we predicted a number of compounds, some of which actually entered clinical trials. One of them was a compound called dutasteride, which is used in reducing prostate size in men of our age. And it turns out in clinical trials that reduced the severity and the length of infection with COVID-19 by about 45%. So there was clearly a rationale for not only finding things that others weren't able to find, but also being able to use those insights to then go back to this huge formulary of drugs that are out there and see whether there was a rationale, a genetic reason why some of those could be expected to work. Because at the end of the day, we so I've been in this industry for an awful long time, and one of the things that you realize is even if we find a novel gene and we get very excited about it and we go on to try and find a new drug for it, even with all of the following wind and all of the luck in the world, it's going to take 10 to 12 years to bring to market. Now, there may very well be molecules out there that modulate that same target, have an impact on its function in a direction that's going to give clinical benefit to patients. And it's out there already. And some of them have been prescribed for decades. So we know they're safe, we know they're well tolerated. So the fastest possible way of getting benefit for patients is to make those connections and to
Mhm.
do it with that genetic rationale I think is super important in the mix here for two reasons. Number one, it gets you to those ideas much quicker. And number two, we can't assume that just because some patients will benefit from this drug, that all patients will benefit from this drug. As I said, there are lots of mechanisms that end up being, in a disease that's called ME at the end of the day. So if only 25
Oh,
and you put them into a clinical trial and you're just selecting patients because they have an ME diagnosis,
right.
the chance of you getting enough signal out of that clinical trial is relatively low. And it may fail, even though the drug will help patients. And that's the bit that we wanted to shortcut. We wanted to say, let's go and find that subset of patients who have this particular mechanism that is driving their disease, and therefore we believe they should benefit from this particular medicine. Do a smaller study, a quicker study, prove that that works, and then then we have a medicine that can help patients. So that was the rationale behind things. It's now 5 years later. We've got five papers out across ME, long COVID, reproducibility. There is a sixth coming. I'll let you ask me about that one because it's very exciting. Um,
Oh, do
Um,
tell,
but
yes.
yeah,
Although I do want to go through some of the basics of your
please.
program,
Yeah,
because it seems like in the past we've kind of been skimming the surface. If you, like you said, if you have this disease, if you have this marker, if you have these symptoms, we're gonna try this treatment, this approach.
yeah,
Uh, but we're not getting at the mechanisms that are causing these things. So it's like it is. It seems like a lot of guesswork because there are multiple mechanisms at play and different. It appears that the same different mechanisms can even cause the same symptoms, similar symptoms. Um,
yep,
so you have this combinatorial analytics platform. That's the start of the
yeah,
process. Can you explain what that?
yeah. So
It's
at the
I
moment,
want to. I also want to say that this stuff is so complex. I
oh,
mean,
it
this is
is.
just. I mean, it's just light years beyond my understanding. But yeah.
It is incredibly complicated, and you're right. Complex is literally the right word for it. Mathematically, it's complex.
Yes. Yeah.
Um, so the way that the way that existing analysis works, the genome-wide association studies that you've reported on previously with with Chris's work and others, they have a relatively straightforward view of disease, and it goes all the way back to Jim Watson and the foundation of the Human Genome Project. They basically thought if we sequence lots of patients' genomes and then we look inside a particular disease, what we can do is find mutations that occur more in this particular patient population than they do in healthy controls. But the critical thing is they were doing it one mutation at a time. So one mutation, one gene. And there's a certain amount of signal that you can find that way, but it is by no means the majority of the signal when you get into complex disease biology. It is for cancer, it is for rare disease, and that's why the tools work really well. And that's why we have a precision medicine industry for those diseases. In complex diseases, as I said earlier, many genes interact with each other. So in biology, if you ever remember one of those biochemical charts on the wall of your
Oh,
biology class, you know how complicated these things are, and they have feedback loops. They literally have circles, but they also have feedback loops. Some of those will amplify each other's effects. Some of them will inhibit each other's effects, and that's where this property that we call nonlinearity comes in. It basically says, 1 plus 2 plus 3 plus 4 doesn't equal necessarily 10. Change those mathematical signs. It might be 1 minus 2 times
mm.
3 times
Ah,
4, and you
okay.
end up with a
Okay.
big negative number instead. And all of that biology, you cannot reproduce that signal from just looking at one thing at a time. You have to study its interactions with other genes, with other factors, the multiome, the immunological, the immune system, and even some environmental factors. So we wanted a platform that could enable us to analyze all of these things. Now, most of the work that we've done in these diseases is looking at those multi-gene interactions. And it turns out that there's a lot more signal there. And that's why we were able to find, in the first ME study, we found 14 genes coming out of the UK biobank population, and we were able to show that that occurred in two different patient populations. When we went into long COVID, initially we found 73 genes in the Sano Gold study. When we moved over to all of us, we were able to replicate 90% of those. So there's consistent signal here. It's just that the existing tools can't find it. And that's really the heart of what we do that's different. And you're right, mathematically, this is crazy complicated. You
Yes.
know, you get up to numbers like 10 to the 40, you know, 10 with 40 zeros behind it, and that's the number of possible combinations you have. There's no computer on the planet, there's no AI on the planet that can deal with that. So we had to find another mathematical way of finding where the signal was that was biologically relevant, and that's what Precision Life does.
Got it. Got it. Well, I see. So, so, um, so you combine all this data together, and then you get these disease signatures, a bunch of disease signatures. Then you divide them up into, you cluster them into communities, and these
Yeah,
are patients with overlapping combinations of signatures, I guess.
yeah,
And they're really interesting things. It's like the innate immune community, the neuronal excitability community, urea cycle, stress signaling, mitochondrial dynamics.
yeah.
Are these patients with are are they quite separate from each other, or are they
It's
or is there a
it's a
melange?
Yeah, more melange.
Okay, okay,
It's a spectrum. You know,
yeah.
my wife says patients are not polite enough to present with only one thing going wrong. Um, and you know, if you have 8 different major mechanisms, patients may have 3 or 4 of those.
Right,
They
right.
may have they may have 1 or 2. That will determine likely the severity and the particular type of symptoms that they're complaining of. And one of the things that we wanted to do, there's another big difference between the way we analyze data and a typical genome-wide association study. That will give you populations level statistics. You know, it will count how many times you see this gene in the whole population, how many times you see it in healthy controls. And that's great for finding that gene. But what it doesn't really do is tell you what are the mechanisms that are likely to be driving disease for an individual patient. And that is the bit that gets me interested because it's much more useful from a clinical perspective to say, this is what's going wrong in this patient. Therefore, if we had some therapies, this is the therapy that that patient is most likely to respond to. So that's the bit that we wanted to get to. And when we divided, when you've seen those communities and we have this mechanistic explanation of what we think is going wrong with them, there's a very natural question to be able to ask, which is, do they show up with a symptom that is consistent with a defect in that particular mechanism of their biology?
Mm-hmm.
So when we did this in ME/CFS, we took genes that were involved in the replenishment of cellular energy supplies, ATP, in the mitochondria. And that was one of the communities. And yes, those were the patients for whom post-exertional malaise was their primary severe feature of their disease. Whereas we had another group who have defects in neurotransmitter biology. They basically can't make the full range of neurotransmitters at the same level as everybody else. And so you would expect that that would be a cognitive impairment cohort. And yeah, those were the patients complaining of brain fog. And there are other groups, circadian rhythm has sleep dysfunction. It also has fibromyalgia, interestingly enough, as a
Oh,
comorbidity.
that makes
There's
sense. Yeah, yeah.
Yeah, insulin receptor. We see high lactate levels in the metabolome, and we see reduced mitochondrial size, which is exactly what you'd expect. So we were able to use the patients themselves to validate the genetic hypotheses that we had, and this kind of clustering that we were doing around mechanism. So that's why we divide things up that way.
Oh, it's fascinating. This was the first time I came across the term mechanostics.
Anyway,
Um,
that's one of ours. We made it up. It's a portmanteau term. Because we're not strictly diagnosing anything. It's there's a difference between a genetic score and
Mhm.
a diagnosis. A genetic score tells you your lifetime risk of having a disease. Doesn't tell you whether you've got it, especially with a pathogen triggered disease, nor does it tell you how severe that is. And a diagnostic that's measuring something that happens when you have that disease is able to do that. So the two are very complementary to each other. The thing a diagnostic doesn't give you is any insight into the mechanism driving
Right.
disease, and it therefore doesn't tell you which therapies are most likely to work for those patients.
But you flip this. Now you get the mechanism, and
Yeah,
you say this is the therapy we think will work in it.
exactly, exactly.
It just makes so much. It makes me wonder why this approach hasn't been taken before. Maybe we didn't have the computer ability or but it seems like searching for the molecular mechanisms behind the disease it's just what MECFS in particular needs because
Yeah,
because we're all over the place. And I mean, we're not all over the place, but we had post-exertional malaise and blah blah blah. But there seem to be so many potential subsets.
it yeah, I totally agree. But it is not trivial to do this. As I said earlier, there are some very, very big computational challenges to solve, and then there are also you have to think about disease from a mechanistic perspective in the first place. And we're very used to that in oncology. We are absolutely not used to it in complex diseases. I mean, how many times do you hear about Alzheimer's and amyloid and ApoE, and that
Yeah, yeah.
being the only thing you need to worry about? It isn't. There's as many mechanisms in Alzheimer's as there are in ME. Um,
Interesting.
yeah,
Interesting.
but
Well, that makes sense too. You know,
yeah. So if you put the two together,
and aging
I
and
yeah. Oh no, absolutely.
yeah.
But if you put the two together, I think you end up with a much more useful view of disease that dovetails into the type of work that's being done at the moment, but also bridges that gap into the clinical community.
Mhm, mhm.
And I think that's a critical piece in a disease that, let's say, has struggled for understanding amongst the many of the medical community.
And particularly the drug community, the pharmaceutical community, and it seems from what I can read, you believe this mechanistic approach will really help us with the drug development. Can you explain how that happens?
Yeah, absolutely. So, one of the things that we've identified. We've published in the paper a variety of different genes where we know there are generic small molecules that are likely to modulate or uh, affect the functioning of that gene in a right way that will lead to a clinical benefit. So the obvious thing to do, as I said earlier, is to find a set of patients who have that particular mechanistic. I keep using the word defect. I don't mean to imply anything negative about that.
We're
That
okay.
is
That's all right.
okay.
We know something's gone wrong. Yeah.
Yeah, something's gone wrong. Exactly. So that mechanism is the driver of their disease.
Mhm.
Now, if we have so I use the word generic small molecule. Why particularly those? It's not because they're the only things that might help that patient. It's that in a small clinical study, you want to have drugs that you can get access to easily, so you can obtain them at clinical grade, and they're relatively cheap. And generic molecules fulfill that. You need them to be safe and well tolerated. Well, if they're generic, they've been on market for at least a decade, so they fulfill that. And then small molecules, because they're easy to give and the dosing is well understood. So it cuts down the number of compounds that we're aiming to trial in the first instance.
Okay.
But it gives us, so we have 9 of these in ME/CFS and long COVID. Many of them actually overlap between the two diseases, and I'll maybe come back to that in a second. Um, but what we want to do, and what we're very actively designing at the moment, are clinical studies to go and find those patients who we believe will benefit, dose them, monitor the changes in their clinical symptoms, and hopefully we will find some or all of them that actually have a benefit for the patient community. Now, it's early days. I don't want to overhype this or get anybody's hopes up, but there is a very strong. This is the same rationale that you would use in a novel drug program. So we're
Mhm.
trying to use the same basic approach, just do it much faster. So
Right.
you've got two outputs from that. The first is potentially a drug that might work. So, one of the targets that we've identified is the toll-like receptor 4. What affects toll-like receptor 4? Low dose naltrexone.
Right.
So
Right. Right.
now you've got a trial, where even if the low dose naltrexone by itself only works in 30% of patients, and therefore, might struggle to demonstrate a benefit in a general trial. If you've picked that patient population and enriched patients who will benefit from that, you may very well be able to find more of a signal sufficient to change the standard of care effectively for that patient subgroup, which will be great. Now, that's the direct impact. The indirect impact is twofold. Number one, you're signaling to the pharma industry that you have a mechanism, a gene, a target in biopharma language, um, which is disease modifying. You've got clinical validation that modulating this target in a certain direction is going to have a clinical benefit. And you're also showing them that you can pick the patients who will benefit from that drug.
Oh, that
Now,
is so
those,
nice. That last one. That's a big. That's the big. That's the big.
so why don't pharma companies pile into long COVID? It's 400 million patients.
Yeah,
It
and
is a
they're
huge
not.
market.
Yeah.
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 Well, they don't for a lot of different reasons, and they don't because it's heterogeneous.
Yeah.
You have multiple symptoms. It's badly diagnosed. You don't have a biomarker where you can track whether patients are getting better or not. You don't know what the mechanisms are. Um, we don't have any good animal models for the disease, so we can't simulate it in an animal system. So you think about what I've just said. You know, Not only are we proving the clinical modification potential and that we can pick patients, but you've already found a way of de-risking that using existing patients. So you using in human trials to do it, but always using safe compounds. And
Right.
so the level of confidence that you have going into a new study in this disease area is much greater that you're going to be successful than you would be otherwise without this information. And I think that, to me, out of all of this, if we can make that successful and give confidence to biopharma companies to invest in this space, will be the game changer. Yes, it would be great to have one or two or three medicines out there that are helping some patients immediately. Of course, it would. That would be awesome. But if we can bring the skills and the economic power of innovation that pharma companies can bring to this, that would be amazing.
Oh, it was that would be such a game. That would be a game changer.
Yeah.
That would obviate decades of MECFS history. Um, um, yeah.
I
Um,
you know I think the community is doing very well. I look at
mhm.
some of the initiatives collecting data. I look at some of our partners. We've got Action for ME and Decode ME in the UK. We've got, as you said, the Complex Disorders Alliance and Metrodora, now Chronicle Bio in the US, doing trials on these things. Um, we've got patient populations of tens of thousands with genomic and clinical histories. That's what's enabling this to work. It's the patient community organizing itself, contributing data, contributing samples, participating in research, and bringing an ecosystem of academic, industrial, small companies like us innovating new ways of looking at these data. And I think it's taken to get, it takes, well, what's the phrase? It takes a village to do this stuff.
Yeah.
It takes a lot of people to do it. You're not going to get there anecdotally with, this with the most respect in the world. One clinician's insight
Mm.
is not going to solve the whole problem. And I, bless them for having an interest in this space and trying to do those things. But you do need a lot more data, and a lot of different perspectives on the problem to actually have a real impact.
It does seem like there's an ecosystem is being built up. You have quite a few partners. I do want to ask, so you
Mm.
found 9 generic drugs that might help, and you're testing them now? Is that correct?
We're designing the tests right now. To be honest, our biggest problem is we are a small company.
Uh-huh.
We are actually studying about 60 different diseases. So we have a number of different areas that we focus on. So in women's health and in obesity and in neuroscience and some other areas. Um, so we need support to run those trials. So we have lobbied and managed in the UK to get funding to support some repurposing studies. Now, unfortunately, we have to bid with partners on that to be part of that program. We have other partners in Australia who are looking to do repurposing studies with us, and some in the US as well. Um, it We're right at the point of well, the trials are designed. We know what we want to do. We know what it's going to cost us. We know how many patients to bring in. We need, we just need the support to go on and do those things.
Right. So you've identified you're looking for super responders. So you feel like you can identify the patients that
Oh
should
yeah,
fit these
yeah. So
with these drugs.
what we've been doing in with the Mellow
Oh, I have
study.
to I'm sorry, I have to let the
Oh,
dog out.
no problem.
Oh, okay. That's good. Okay. Just tear the door apart.
Yeah, so what we've done with the Mellow study, which is ME long COVID, with Chronicle Bio and the Complex Disorder Alliance. So those are the new names for Metrodora Institute and Metrodora Foundation.
Oh, okay. Okay.
We recruited 400 patients with ME and/or long COVID, and we've been testing a buccal swab test where we can run their genotyping and then identify the mechanisms that are driving their disease, and whether those are really high, average, or low for a patient with whichever of those diseases they believe they have. And what that's allowed us to do is to get a level of confidence that we can identify patients using a test which can be delivered at home, that can find those patients who have a specific mechanistic signature, and that we can use to do the recruitment. So we can either do it from an existing sample that, if they've been enrolled in a particular clinical trial previously and have some leftover sample, we can do that. Or if there's a registry of these patients being treated at a particular center, we can send them a test and see which of the trials uh, they may be suited for.
In the in the so the Decode ME trial,
Mm.
you got a grant to use
Yeah,
decode ME data, and
yeah,
I
yeah, I
assume
did.
you're getting data all the time,
So
or
we've, yeah, I mean, Decode ME now has actually finished. So the funding period of that ran out at the end of August, which is why you saw, kind of the paper that came out from Chris and the Decode consortium, or sorry, the preprint, I should say. Um, we've got access to a significant fraction of that data. There's a certain number of patients who didn't consent for commercial research, and we had some other QC issues. But basically, we've got about 11,500 patients worth of data coming out of that study. And that's great because that's easily enough for us to do these types of studies with. So yeah, we've got that data set. We've actually got 4 data sets that we use. So UK Biobank is a great source. We also got the Sano Gold study, which was long COVID, and we also got access to all of us, and we've replicated both the the long COVID data in all of us, and then we've replicated the UK Biobank findings in Decode ME. So we've done a lot of cross validation across the
right,
different data sources. So we're
right.
we're getting very confident of our results right now.
And is there is precision life it's mostly focused on genetic data. Are any other types of data being included in these
It
studies?
it it is in these diseases because that's the data that's been collected
Uh-huh.
alongside primary and secondary care EHR, the electronic health record data. Um, in other studies, we've looked at proteomics, we've looked at epigenetics, we've looked at metabolomic data, immune phenotypes, microbiome, environmental, epidemiological. So there's a lot of it depends on the disease. I mean, if you take something like diabetes, there's or Alzheimer's we were talking about earlier, there's lots of social determinants of health that come into those kind of diseases, and they're big, powerful effects more so than they are in ME and long COVID. What we worry about in ME and long COVID is obviously the ancestry, the diversity across different ancestries.
Mm-hmm,
So
hmm.
we went looking when we did that reproducibility work. One of the challenges is, in the UK, most of our data sets are 94% white European.
Right,
So it's been very difficult to translate those results or be confident in translating them out into different ancestries. So we did that particularly in all of us, and there's good reasons why this should be the case, but we were very happy to see that we were reproducing 60 to 70% of the signal in Black, African American, and Hispanic
oh,
Latino populations. That is lower than the sort of 80 to 90% that we were getting in white European, but nonetheless, it's much, much better than you would get with a traditional kind of GWAS or polygenic risk score approach.
mm.
So,
Oh, okay.
yeah.
So yeah, similar things are happening in all the races.
Well, we
It
can we
appears.
can certainly find mechanisms that are associated with the disease across all of them, and a
Right,
significant proportion of them, which is sort of to the point because if you're going to choose a medicine, you want it to work in as many patients as you possibly can.
right, right. So in MECFS is multi-omics data being incorporated, or
No, not
is it gene expression right now?
it. It's genetics, so genotype
Phenotype,
specifically.
uh-huh.
Yeah,
Um,
and we have done some gene expression work, but the challenge with that is, as you know, gene expression, epigenetics, proteomics are all very tissue specific. And of course, we have a lot of different tissues involved in these diseases. And so
oh,
getting consistent signal across those is really challenging to do. Bloods are about the only consistent tissue that you can you can collect in that way,
Mhm.
but they may not be directly correlated with the level of signal that you would see in the primary tissue. So, if in the immune system, well, yeah, I mean, if you have tissue specific effects, they may show up in blood, but they may not, and
Uh-huh.
that's another level of complexity over and above, the genotyping studies. What we are hopeful is that the genotyping will allow us to subset patients and say, look, this is the driver of disease. It is occurring in this particular tissue or this organelle. These are therefore the types of studies you should do. Let's go collect a data set which is more specific to that patient population and goes a lot deeper with metabolomic or uh, expression or proteomic or whatever the multiomic signature is.
Well, what it seems like, I mean, when I look at the list of is this list of mechanistic groups, I guess, or mechanisms: post-viral, immunometabolic, vascular, autonomic dysregulation, neuroinflammatory, cognitive, hypometabolic, mitochondrial, gut immune barrier. Those are all prominent aspects
I
of
I
ME/CFS.
I have to tell you a funny story here.
Uh-huh.
Um, uh, I hope Chris won't mind me mentioning this. So Chris Ponting, um, when we started this work, um, we only had UK Biobank to go from. So we were trying to go from COVID to long COVID, but nobody had collected a long COVID data set. So the only one that we could find was ME in UK Biobank. Now, we knew nothing about this disease. I mean, literally nothing about this disease. I'm going back to probably the beginning of 2022 here. So we did what we usually do. We did a hypothesis-free analysis, and we pulled up that picture that you see with 200 SNPs and the communities and connection to mechanisms. And to be absolutely honest, we almost threw it away because we didn't know whether it was any good or not. And there weren't we weren't seeing the same set of hits that we would have expected
Mm.
for the number of patients that we'd included in the study. Usually, frankly, we get many more SNPs associated. Um, but we passed it in front of Chris, and Chris, Chris immediately accused us of confirmation bias,
Oh,
saying,
oh,
"Oh,
really?
these are you've chosen these mechanisms, and this is what you're
Oh,
seeing."
oh,
At which point we had to tell Chris that we had no clue what was involved in this disease, and
oh.
if we had found things that replicated, phenotypes and mechanisms that were thought to be associated, that was a very positive thing. So together we came to an understanding that perhaps there was some very useful signal here, but it was a very lucky coincidence that we had, such an experienced KOL, scientist, experienced in the disease, working with us, because it is one of the challenges, particularly when we go into these areas for the first time. We may be able to find really interesting signal, but it does not make us disease experts. And
Right,
that's
right.
a very interesting situation to be in. We've solved this now by obviously getting a really great team of experienced scientific advisors alongside us in each of the studies that we do. But we are dependent on them to tell us the credibility of the findings that we're generating.
Right, right. But how validating. It just all popped up. He's like, this is too close. This is too
Yeah,
close. Yeah, yeah, yeah.
exactly,
I know what you're
exactly.
doing. And
Was
good for him. Good for him too.
Yeah.
Yeah. Can you say anything about the 9 generic drugs that you're interested in?
Yeah, I mean, I mentioned one of them, the low dose naltrexone. I think we're really interested in. We've got others that are involved in things like blood pressure, which we think might be more amenable to POTS populations, for
Hmm.
example. Um, of of the nine, five of them are FDA approved, so very easy to test in the US. Um,
Are these drugs that are currently being used in
oh yeah,
ME/CFS? Uh-huh.
no, well, not so much some of them potentially in MECFS. Yeah, so there was one of them, and I can go back and check. I'm not avoiding the question. I just can't remember exactly what what the drug was. There's one that has been used, and it's one of those frustrating ones that anecdotally seems to work in some patients and doesn't in others. Well, that's
Perfect.
exactly what we would expect, right?
Right.
But for that reason,
Right.
it's been thought by the clinical community perhaps not to be particularly interesting. And I think you see this, if you go back to the Treat Me or Treat ME
Hmm.
study, I think you're seeing, aside from those obvious things like pacing and IVIG, for any given therapy, you see a typical pattern of response that is going to be 20 to 30-ish
Right.
percent that
Right.
it
Right.
benefits, a similar population who seem to get some level of harm from it, and a whole bunch of people in the middle. Um, Obviously the intention is to try and be more specific about matching those together. Um, there are a number of other, so we had 13 repurposing targets, and um, when we looked across ME and long COVID, so one of the things we haven't talked about is there is a very obvious genetic overlap between those two diseases, and we've shown that we showed that in our original long COVID paper, which was October
How,
20.
yeah, how significant is that? So people have a certain genetic susceptibility to long COVID, and people have a genetic susceptibility to ME/CFS.
Mhm.
And of course, long COVID is associated with the coronavirus, and ME/CFS could be associated with many different triggers.
I think
Uh,
that's all true. The question is, does that do the mechanisms that trigger one almost inevitably cascade into the other?
uh-huh,
In
mhm.
other words, is ME a sort of end point for
Right.
something that starts through long COVID? And it looks like the answer is probably yes.
Hmm.
At least for some of the mechanisms, so
Okay.
how significant was it of those first 14 genes that we found in ME or MECFS? Sorry, I do think of them as different diseases, but technically we were studying an MECFS population in UK Biobank. So of those 14 genes that we found nine of them in long
Oh,
COVID.
that's pretty
So,
good. Yeah.
and then when we did the replication study for long COVID, seven of those nine reproduced in the all of us population, and that the other two we couldn't test because they weren't actually in the data set, in the all of us data set.
Okay.
So our presumption is that pretty much all of those have survived or will survive when we get better data. What we're also doing.
Wait, well, can I ask? Can I
Oh
ask? When
yeah,
you're looking at all of us, I mean, are you looking at the people who've been identified with ME/CFS or long COVID? Is that how you
long COVID in this
got it?
particular
Got
case.
it.
That
Okay.
was,
Okay. Okay.
yeah.
Okay. Oh
So
yeah.
the challenge is the coding in these, in these prospective studies. The coding that you get is, essentially coming from claims data, and how badly,
Oh, oh. Of
how
course.
inconsistent
Yes.
those
Right.
are. So,
Yeah. Yeah. Yeah.
um, we we've we actually restricted the number that we put in the study very severely to those that we really, really thought we could be confident had
Yeah.
disease. And we excluded a whole bunch who very probably had the disease, which disadvantaged us, but still, we feel, more comfortable if we have confidence in the diagnosis. Um, so yeah, we went on and did that. But we've come a long way since those initial, that 9 out of 14 number and 7 out of 9 replicating. So of course we found 73 genes associated with long COVID.
Right.
Um,
Right.
what we're doing now is analyzing decode ME.
Hmm.
So I can tell you that we have three-figure number of genes associated with decode
Whoa.
ME.
Really
Yeah, and so this
interesting.
is
Does
it's
that
going to
make
be a
sense that there would be more because it's more heterogeneous?
I think it makes sense because our sample size is larger.
Okay.
So
Okay.
I think they're equivalently complex as disorders, and they have similar kinds of mechanisms as we've established. But we simply have more numbers. So we have about 11,500 patients in Decode ME, whereas we only had a couple of thousand in long COVID. In fact, less than that. It was, yeah, well, it was about 1,200 by the time we'd gone through this. So we expect to see more signal just because there's a level of statistical validation that needs to happen, and that's easier with higher numbers. Um, but yeah, so there will be a new preprint um, published or uploaded in about 3 weeks' time,
Okay. Okay.
where we're going to disclose these findings coming out of Decode ME. So this kind of builds on Chris's and the Decode ME preprint. I think they found a number of loci.
Mhm.
They be, just because of the low numbers and the different way that the statistics work, they weren't able to replicate those in the other data sets that they used.
Right. Right.
I don't think that that's because they're wrong at all. I think it's just because they didn't have sufficient sample size to be able to do that.
Right. Right.
We need a lower sample size. So, the way our method works, we need fewer patients to validate a signal. And we are seeing the same things that Chris saw and
Oh,
a whole bunch more as well.
and
Um,
let me remind everybody that you're using this combinational analytics thing to
Absolutely.
map all the interactions and how everything is working together to come up with these signals.
Yeah, exactly. So
Wow.
yeah, it lets us it's almost it's almost like turning the lens round on a microscope. It's just getting to that next level of resolution, if you like. That's
Right.
the way we kind of think about it. But it is very encouraging that we see exactly the same signals. They're reproducing across data sets. They're reproducing as we expect between diseases as well. And so when we start to think about, what are the mechanisms we choose for repurposing, I think coming out of this next study, we're going to have a lot more candidates, and we're going to be able to say, yeah, we can, we've now replicated in 3, 4 different data sets, and we have really high confidence that we should be able to find drugs that modulate these targets in a way that's going to be beneficial for patients.
Hmm. So the next step is the mechanostics step.
You got to get that
That's
test
got to
in
come next.
because yeah,
Uh-huh.
you got to get the test in. That's so we've with the Complex Disorder Alliance and Chronicle, we have done this on 400 patients. We're in the process of returning results to those first 400 patients. Um, they're looking encouraging. I don't want to overhype this at all. There's a long way to go between here and there. This is still research, but they are looking encouraging that we're going to be able to find patients who, in principle, ought to be able to benefit from particular therapeutic approaches, and that's really what we need to get the IRB, the ethics approval to run a study inside some of those patient subgroups, and that's the necessary next step alongside the funding to go and set those clinical studies out.
Well, that's great. That's great. It reminds me that Ian Lipkin is doing a big
Yeah,
GWAS study, I think, and
yeah,
that should only add to the add to,
I think so.
mhm. Did
Yeah,
anything
sorry.
did anything surprising pop out once you got this bigger population and you have more mechanism or me we haven't gotten to the mechanism part, but you have more communities, I guess, or disease signatures.
I think there's a bunch of really interesting things in there. I mean, what I would, the surprising thing is that we got so close to as many of the weird and wonderful mechanisms that we see coming out in the symptoms of patients. It's unprecedented for a disease like long COVID to have 200 symptoms, affecting so many different parts of the body.
Right, right.
And that totally unexpected from a pathogen triggered disease.
Hmm.
And as I said, right from the very first study, we find blocks of genes associated with particular mechanisms that really get into the heart of those. I think one other thing I would say, and this hasn't been published yet, but we will be publishing on it. Um, we have also seen overlap with diseases like fibromyalgia, for
Oh, that's
example.
great. That's great.
So
Fibromyalgia really
yeah,
needs some help.
well, there is a
Interest. It needs
preprint.
interest. Yes, yes.
Yeah, I mean, there is a preprint out of a GWAS study that has, I think, found 8 loci.
Hmm, hmm.
Um, we see 6 of those in ME/CFS.
Oh, great.
So, now you're getting into which clinical label do you put on this
Right.
patient who's presenting to you? Do they even remember an infectious trigger to their disease, or have
Hmm.
they presented with
Right,
something
right,
that looks so similar to fibromyalgia that that's the label you're going to put on this?
right.
And, again, it's partly about mechanisms, it's partly about the observation of clinical symptoms, because that's where your clinical labels come from. But what it doesn't get to are what are the commonalities of mechanism that are driving disease across all of these indications. And we
Hmm.
would love to be able, if we do demonstrate that we have something that is effective in ME, to be able to show that, that will also benefit long COVID and potentially, if it's the right sort of mechanism, potentially something like fibromyalgia as well.
In the end, you should really be creating new disease labels, shouldn't you? Uh,
That's what we've done
this
in
is an
cancer.
autonomic blah 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 disease.
Yeah. That's what we've done in cancer. We've turned
Uh-huh.
cancer from a disease that is defined by the site of onset. Breast cancer. 50 years ago, we had one form of breast cancer, and it was called breast cancer because it occurred in the breast.
Right.
Now we have at least 120, and it's defined by, the hormones and how responsive to manipulation
Oh
of
my
hormone
goodness.
levels they are, which particular mutations are driving, the formation of oncogenic tissue. Um, and we have medicines for many of them. So it becomes a palette of molecularly defined subtypes of a
Right.
disease, and we choose the therapy that matches the subtype, where we have effective therapies for them. That's what bringing precision medicine to complex disease is all about. It's about understanding these broad mechanisms and finding therapies that work against them. And don't worry about, yeah, many of these labels come from Victorian or Edwardian clinical observations.
Mm.
They're the language of medicine actually is not helping us find more specific therapies that will actually help patients. And so breaking it into a more precision approach, a more mechanism focused approach, I think offers a lot of potential for actually finding therapies that help patients.
Well, I wish the Recover Project had talked to you first, because they've been doing the exact opposite. It seems to me, um, but hopefully
Yeah,
that's that'll be changing. Uh,
I hope, a lot of this comes down to what do you believe is possible? And, you have to take
yeah.
your hat off to the pioneers who went out and got funding to do the big studies around genetics when people were saying, well, what on earth does genetics have to do with this disease? It's pathogen mediated.
Hmm.
You get it after you've had an infection. What's good genetics got to do with that? And the short answer is not 100%. It determines your propensities and your symptoms and the mechanisms and the drugs that you're likely to respond to. But there's a whole bunch of other factors involved in that as well. So nothing is ever as clean as you would like it to be. But without those pioneers going and collecting those data, we wouldn't be able to do what we do. And the other side of that, of course, is without patients willing to share their data and make it available for research. Particularly trusting a company to do that research. We wouldn't have these kind of findings in any of the diseases that we study.
It seems like with your combinational approach, you're saying genetics actually makes more of a difference than we thought. Uh,
Yeah, I do. You see this in a lot of diseases. So we're just on the point of publishing another paper in endometriosis,
oh, good,
which is a
good.
similarly complex disease. Horrible from a clinical perspective. Women take 8 to 10 years to get prescribed, to get
Yeah.
diagnosed, and there are no effective medicines out there. Hormonal therapy, but you can't use that if you want to get pregnant, and it has lots of side effects. Um, and you start to look at these disorders, and you start to break them. You start to break them down, and you end up, with a very different kind of appreciation for what is similar and what is different inside disease versus normal kind of tissue. Um, and yeah, we see regularly, um, a lot of things showing up inside diseases that are, well, let's take endometriosis. The best meta GWAS that was out there, which had 60,000 patients and about 700,000 controls, found 42 SNPs. Together, those explained about 5% of the disease biology,
Right, right, right.
whereas it's known that endometriosis is 50% heritable.
Mm,
So what's
mm.
that gap? It's genetic because it's heritable. We can only see 5% of the signal. That is, 1/10 of the genetic component of the disease. So where's the other 90% of that genetic signal? Some of it is in these combinations, and
Mhm.
that lets us see much deeper. So instead of 42 SNPs, we've just reported on about 3,000 SNPs that we found, or we will do. Sorry, that's going to get published well, we've put the preprint up for that one. So there is a lot more signal there in genetics. It is not the only contributor. I mean,
Right.
endometriosis, 50% heritable, so 50% is not. And there's
Interesting.
something else going on. It's absolutely true in ME and long COVID as well. Similar kind of issues.
Oh, my goodness. So, so with regard to the, so you have these papers coming out. You have a much bigger study coming out with
Yep.
more signatures, and that'll be very interesting. And you're going to work on translating that into the mechanisms, and then looking for drug biomarkers, and then drugs that fit the mechanisms, and being able to track them efficacy with biomarkers, if I have that right. And so what is going on with clinical so you but right now you don't have the funding to do the clinical trials,
Yeah,
is that correct?
we're
You're
we're
working on
applying
that.
we're applying for funding
Got it.
to do that. Unfortunately, Precision Life is a fairly small company. We're very good at doing the analysis side of things, but it does get expensive very quickly when
The
you
clinical
need to do
trials, yeah.
clinical studies. Yeah. Uh,
Even if you're doing super targeted, yeah, yeah,
yeah.
yeah,
I
yeah.
mean, these are not in the scheme of things, these are not super expensive.
Hmm.
It's the sort of thing that could easily be funded by foundations or patient charities, even if there was sufficient interest in doing so. But it is beyond our means to fund all of that
Right.
from our existing resources.
Got it. Well, this has just been a fascinating talk, and I've learned so much, and uh, that I it sounds like the future seems bright, um,
Look,
for
it's
you and hopefully for us. Yeah.
well, we hope so. It's early days. I don't, there is always a long way from research to having a clinical impact. But
Right. Right.
what I
Right.
do believe, and I do feel confident saying, is that we understand the disease much better now, and will do, particularly with the next set of studies that come out, than we ever have in the past, and that should give us um, it gives us a leg up in all of the next set of studies that will go on to clinically validate all of these different mechanisms and show their relevance to the disease. And I think that can only be a positive thing.
Absolutely. Absolutely. Well, I wish you, of course, I wish you the best of luck, and
Thank you.
I'll be eagerly looking to see what shows up.
Yeah. Well,
Yeah.
we're really thankful for the community's interest and participation. And I think there are, you know, the next few years is going to be really important for the community to come together around some of the trials that will inevitably come out of our work and everybody else's work doing these kind of things. The more support we get from the community, and you've been a great advocate and organizer of that. Um, the quicker that research will go. We, at the end of the day, we need patients in order to make this work. So we're very thankful for what they've done so far, and very hopeful that we can incorporate them in the research in the future.
Well, I think you can be guaranteed of a pretty eager patient community.
Well, yeah, yeah. Well, that's why I don't want to get ahead. It is still research, and there is a translation step that needs to happen into the
Yes,
clinic. But
it is very complex, and I guess the more complex the analysis, the more careful you have to be at winnowing out false positives, or what isn't real from what's real. I imagine
yeah,
that's quite a process that you have to go through.
it it is, but that's what the clinical trials and studies are for. So
That's right. That's right. Okay, well, thank you again so much for your time, and good luck. Yeah,
thank
it's
you very
been
much.
it's been fun. Yeah.
We'll keep you in the loop. We'll send you a copy of the preprint when it's of the next study when it's up.
Oh, good, 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 good. Very good, very good. Well, thank you so much,
All right, Steve. Yeah,
Steve. Yes,
yeah.
a nice suit, by the way. When, when we started this, you said, "I don't usually dress like this. I just went to a swanky event, and so, you know."
I didn't. Yeah, it is a little unusual. Let's put it that way. I had to iron a
Okay.
shirt.
All right.
All
Okay.
right,
Well, thank you again. Bye bye.
lovely to speak to you, Cort. Bye.
Yes, yes. Bye bye.